Hello and welcome to this chapter on microbial growth. This chapter is intended for my college students, but of course anybody can watch this and hopefully gain some kind of benefit from it. So in this chapter, we'll be looking at different factors that affect the growth of bacteria and other microbes, and we'll look at chemical factors, physical factors, what do they require for their growth, those types of things. This is a three-part lecture series. Not too complex, it's pretty straightforward and easy to follow, so let's get started. So when we talk about microbial growth, we're not talking about the increase in the cell size. What we're talking about with growth is the increase in the number of the cells. So as time goes on, the number of cells increases. We talk about exponential growth, and that's how bacteria do grow. Their numbers will just continue doubling and doubling and doubling until the environment cannot sustain them any longer. So the term growth refers to increase in the number of the cells. And two terms that I want you to be aware of in relation to growth are colony and population. So a colony would be a group of hundreds of thousands of cells. If you were looking at a petri plate, which is the plate that you grow the bacteria on the agar, which is the growth substrate, you would see areas of growth. Well, there would be little dots where there would be bacterial cells. One of those dots on an agar plate, on a petri plate, would represent a colony. And in that dot, there would be hundreds of thousands of cells. So they're very, very small, and it's easy to just pluck up hundreds of them very easily right off the agar and put them onto a slide and then observe them. A population of cells would be a group of billions of cells. So those are the two differences in them. Like, for example, on a petri plate, there would be a population of bacteria. There would be literally billions of cells on that petri plate of the bacteria growing on it and thriving, because their nutrients are ideal, the optimal temperature, everything is perfect for them, so their numbers can grow to enormous quantities. So physical requirements for their growth, we'll look at a few of these. We'll look at physical first, and then we'll turn to the chemical requirements that are necessary for their growth. In regard to temperature, there are 5 primary groups. And they go from the cold to the hot. So those are psychrophils, psychotrophs, mesophils or mesophils, thermophils, and hyperthermophils. So all of these are related in some way to their temperature and what temperature is optimal for them to grow in. And so you can see, and I don't know if you knew that bacteria can grow in temperatures as low as 15 Fahrenheit. So that's well below the freezing point, and they can actually survive there. Now, there are very few species that are in that category, just as there are very few species in the hyperthermophil category over the boiling point of water. So these would be the bacteria that can live in thermal vents at the bottom of the ocean floor or in hot sulfur springs, say for example in Yellowstone Park, where the temperatures are very high, around that 200 to 250 degree range. Most bacteria, however, are right in the middle at the mesophil group, and meso, that prefix means middle. So, and that's kind of where the temperatures are on average around the Earth. It's kind of in that middle temperature range. That's where most of the people live. That's where most of the organisms on Earth live. They live in that middle temperature range. And this is all based on enzymes. How do their enzymes function? Temperature is a necessary requirement for enzymatic function, and so this becomes an important physical requirement because temperature is an important factor for enzyme function. And this is just a graph showing that they have optimal growth temperatures. They peak at a particular temperature and then they decline, and then the next group comes in and it peaks at a particular temperature, and so on. So this is showing a graph, and you can see temperature in Celsius. And if you look at the mesophils or mesophils, meso sounds kind of funny, but that's how it's supposed to be pronounced. But many people say meso as well. But anyway, sorry, off topic. But at the mesophil category, you'll notice that it peaks at 37 degrees Celsius. Well, do you know of any organisms that have a temperature, a body temperature of 37 degrees Celsius? That would be us. And so we become the perfect incubator for the mesophilic bacteria that are thriving at 37 degrees Celsius. And you can see thermophils at a higher temperature, and then hyperthermophils as well. So this is all related to optimum enzymatic activity. What is the perfect temperature for their enzymes to function? And as you know from the previous chapter, chapter 5, we talked about enzymes. They are required for all biochemical processes that occur in a cell. And so that enables life to go on, that enables biochemical processes to actually occur. You have to have the enzymes. And in order to have the enzymes functioning properly, you've got to have the perfect temperature for them to thrive in. This is just a thermometer showing the temperatures where they're going to grow. And so this is more of a practical graphic here that's showing that center yellow area called the danger zone. It sounds so ominous, the danger zone. That's the rapid growth of bacteria. That's the zone in which their growth is going to be exponential. And in that danger zone, I guess this is what makes it so dangerous, is that they're producing their toxins. And then you go above or below that, and you see that the rate of growth decreases at warmer temperatures and at cooler temperatures. So in terms of Fahrenheit, the danger zone ranges from around 60 degrees Fahrenheit to 130 degrees Fahrenheit. So that's the area where many bacteria are thriving and living and growing exponentially. Then you get down to the refrigerator temperatures, where at the refrigerator temperature, it doesn't kill the bacteria, but it really slows down their metabolism. It really slows down then their rate of division. It renders their ability to produce toxins pretty much useless. They aren't able to produce their toxins very well. And then when you get down below freezing, for many species of bacteria, there's no significant growth at all. Now, freezing does not sterilize. Endospores can survive freezing temperatures, and then whenever say the food, you take it out of the freezer and then it comes to temperature again, even if it's in a vacuum sealed container, bacterial growth will revive once it gets back up to the optimal temperature range. So this is just showing basically in terms of our food storage and what temperatures we should be storing them at in order to reduce their growth. pH. Now, pH is kind of a chemical, physical requirement. We're going to look at it in terms of physical requirements here. They grow between a pH of 6.5 and 7.5. And if you know your pH scale, that's right about neutral. 7.0 is exactly neutral, where the H plus ion concentration is exactly equal to the OH negative ion concentration. Very few bacteria grow below a pH of 4. There are some that do, and they're called acidophils, and they love that acidic environment, but very few grow below that because a low pH will denature enzymes. Enzymes denature, cells can't survive. So this is why pickling food prevents spoilage. The fermentation produces acid. So when we pickle foods, we encourage them and put them in an environment in which they'll start producing lactic acid. And so we use particular species to do that, like lactobacilli species of bacteria that will convert the carbohydrates into lactic acid. That lowers the pH, and then that prevents the spoilage of food. So normal spoilage bacteria cannot tolerate those lower pH temperatures, please. They can't tolerate the lower pH ranges where they are normally surviving in the neutral pH range. And so they get into that environment, they die, and then they can't produce toxins. We don't get any kind of food poisoning or illness from those foods that have been pickled. Molds and yeast grow between a pH of 5 and 6, so they're thriving in a pH range that is quite a bit lower than the optimal bacterial pH range. And so, for example, what typically rots are fruits and vegetables. It's mold does, not bacteria. Because fruits and vegetables typically have a lower pH, they're more on the acidic side, and so molds can get onto our fruits and vegetables, and they can actually thrive on them, and they're the ones that cause the spoilage of those types of foods. And there are a very small number of bacteria called acidophils. They can survive in extremely acidic environments as low as a pH of 1, which is about as low as you can go on the pH scale. And there's a small number of species that can survive in that pH value. So we'll look at water and in relation to osmotic pressure. And so we learned about and talked about in a previous chapter about the different tonics, isotonic, hyper, and hypotonic. Hypertonic environments increase the solute concentration, so you have a higher amount of solutes on the outside of the cell, and what that causes then is water to osmose out of the bacterial cells, which leads to plasmolysis, and so they can't survive in those conditions. So we took advantage of this when we preserved foods by foods were meats, for example, were submerged in salts before we had refrigeration as a common method of food preservation, and so the meats would be submerged in salt for weeks, and then they could be taken out and they would be fully cured, and then bacteria could not grow on them because of the hypertonic environment that's created as a result of the salting of the foods. Hypotonic environments decrease solute concentration and cause the cells to lyse. So we have a very low solute concentration compared to, on the outside of the bacterial cell compared to the inside of the bacterial cell. So water will osmose into the cell and the cell will burst. Now typically bacteria, they do live in hypotonic environments, but because of their rigid cell walls, they don't burst because the cell walls can resist osmotic pressure. But we can use this to our advantage with our antibiotics. Whenever penicillin was discovered in 1928, we realized that it does kill bacteria. Now then we didn't know exactly how it was working, but later we found out that what it does is it breaks apart the peptidoglycan of the cell wall. Water then, when the cell wall is, the integrity of it is broken down, water can rush in, it can osmose into the cell, and the cell will lyse. Extreme or obligate halophils require high salt concentration in order to grow. So obligate halophils, obligate means they're obligated to be in a salty environment. And the prefix HALO, that refers to the halogens, which refers to the salts on the periodic table. And then PHIL means to love. So they are obligated to love salt, basically is what that says. They need a salt concentration of about 30%. That is approximately 10 times saltier than ocean water. So we're talking about very limited number of species, very limited number of geographic areas where they can actually live. We're talking about the Dead Sea or the Great Salt Lake. Is where those obligate halophils would survive. Facultative halophils tolerate, but they don't require high salt concentrations. So they can live in a salty environment, now not extremely salty like 30%, but around 10%. So Staph aureus is probably the most common facultative halophil. It can survive in salty or sugary foods, and then this is what leads to food poisoning, or I guess a better term would be food intoxication, but we use the word intoxication for being drunk, so that sounds kind of weird. But technically that's what it is. So what happens is other bacterial species cannot tolerate the slightly salty environment or the sugary environments of particular foods that we consume. Staph aureus bacteria get into those foods. They don't have any competition, and so they can multiply and divide. They'll produce their toxins. Those toxins then get consumed by individuals, in which the food has had time to sit, and then the toxins have had time to accumulate, and then that food gets served to individuals. They consume the food with the high concentration of Staph aureus toxins. The Staph aureus exotoxin is a super antigen which causes a cytokine storm, and it causes violent reaction, lots and lots of vomiting. So the body is trying to remove the toxin through the process of vomiting up and vomiting repeatedly and and very forcefully. So those are what are Staph aureus and others. Those are what are called facultative halophils. They tolerate it, but they don't need it. This is just showing plasmolysis here. You got the cell wall remaining intact, but the cell membrane, the plasma membrane, shrinks in due to the loss of water osmosing out of the cells. The bottom picture, I couldn't find a great micrograph of a bacterial cell undergoing plasmolysis, so I have a picture of an aquatic plant called Elodia, and you can see the cell walls are clear and they're still remaining intact. They're still that rectangular shape, but you can see the plasma membrane has shrunken in. And the reason why it's green is because the chloroplasts get trapped in the cell membrane and then get drawn inward to the center of the cell. This would be lysis. So here's the E. coli after exposure to an antibiotic. It's spewing out its cytoplasm, and all that black stuff, that would mainly be ribosomes that are coming out of the cell, along with some of the nucleic acids as well. So this is what bursting looks like. That's exciting. So let's look at some chemical requirements. In other words, we're going to look at the elements. Carbon, number one, most important and abundant element. It's a backbone of all organic molecules, and it serves as an energy source as well. Chemoheterotrophs, so these are organisms that must consume, they must get their carbon from other sources. They're not able to make it themselves. They're using organic carbon sources. Autotrophs will use carbon dioxide to get their carbon. So, like for example, with photoautotrophs, they're taking in carbon dioxide, like algae, for example, and they're splitting the carbon dioxide and using the carbon to build up carbohydrates, mainly glucose, and then using the glucose to build other compounds from there. Nitrogen is used to make amino acids. Proteins, of course, because amino acids are the building blocks of proteins, DNA and RNA. So in DNA and RNA, those are nitrogenous bases, the adenine and the thymine and so on. Those are nitrogenous or nitrogen-containing bases that make up the rungs of the DNA ladder and the steps on the RNA. So most bacteria, in order to get their nitrogen, they decompose proteins. So they break them down into their smaller constituent parts through the use of their enzymes, of course, and then they can obtain the nitrogen that way, and then they can use that nitrogen to rebuild up new proteins for them and make new nucleic acids for them. So this is what life really is. I mean, we're just taking organic compounds and we're consuming them, and then our body is breaking it down and then converting it into a form that's usable by us or whatever organism happens to be consuming it. So whenever bacteria, when they degrade, say, this is kind of unpleasant, but say roadkill, and there's quite a bit of it on the roads of Pennsylvania this time of year, they're breaking down those proteins that are useless to them. So they're going to break them down into their smaller parts, and then just rebuild them back up again into compounds that are suitable for them in order for bacteria to survive. And we do the same thing. The foods we consume, they're kind of useless in the form that they're in, but they provide us the energy that's necessary to build new compounds and molecules within us using them. So everything just gets recycled. Some bacteria can use ammonia or NO3, which would be nitrates. So they take those from the soil, and they can use those compounds, break them down through the use of enzymes, obtain the nitrogen, and then use the nitrogen to build up their own proteins, etc. A few bacteria, and there aren't many, it's a very small number of them, they use nitrogen gas from the atmosphere through a process called nitrogen fixation. So the atmosphere that's all around us right now is about 78% nitrogen, and that nitrogen is pretty much inert for nearly all organisms. Like for us, we just breathe it in and we breathe it right back out again. Some of it passes through actually goes down our esophagus too and passes through our digestive tract, and we get rid of it at the other end in gas, but it doesn't get picked up by anything in the body. The blood doesn't react with it, it doesn't get picked up by hemoglobin, and so it just passes right through us. Well, these bacteria can actually use the nitrogen gas, the abundance of it that's all around us, for their own metabolic processes and building up their compounds, which is very handy for them. I mean, they've got pretty much an unlimited supply of nitrogen available to them just taking it from the atmosphere. And there are very few organisms that can do that. They're very important in an agricultural aspect, where these bacteria grow on the roots of plants. they fix the nitrogen and they make nitrates out of it, and then the plants can get their nitrogen from the compounds that these nitrogen fixing bacteria are producing themselves. They produce more than they need, and so this becomes a mutualistic relationship between the nitrogen fixing bacteria and the plants, and it's a wonderful relationship between the two of them. Sulfur is used to make amino acids, thiamine, biotin. These are the B vitamins. So they become, sulfur is necessary to make those category of compounds. Most bacteria decompose proteins to get their sulfur. So proteins are held together by sulfur bridges, sulfur-containing compounds that bridge the proteins together and enable them to have these complex shapes and hold them together. Well, when they break down the proteins, they can obtain the sulfur. Some of them will use sulfate or hydrogen sulfide in order to get their sulfur as well. Phosphorus, which is necessary to make DNA and RNA. It's needed to make the backbone of those compounds. There's a sugar and a phosphate backbone, and so phosphorus is needed there. It's needed for adenosine triphosphate, and it's needed for the phospholipids in cell membranes or organelle membranes. So phosphorus is needed in a variety of vital ways. And phosphate is a source of phosphorus. So phosphate is locked up in the rocks. The rocks become eroded. The water dissolves it and releases the phosphates over long periods of time. The phosphates get deposited into the soil, get taken up by plants, and then this is the basis of the phosphorus cycle. All of these elements are being cycled through the environment. The nitrogen cycle, the carbon cycle, there's a sulfur cycle. So all of these are cyclical processes that are occurring in the environment, releasing these elements and making them available to organisms. And then trace elements, trace meaning needed in very small amounts. We don't need many of these. Primarily, and this would be, I keep saying we, but this is supposed to be from the aspect of bacteria, but we need trace elements as well. For bacteria, it's primarily iron, copper, molybdenum, and zinc. So those are the four that are needed in small amounts. We need small amounts of these elements as well. Too much can be a bad thing, and so that's why only very small amounts of them are needed, and most of them are used as enzyme cofactors to enable the enzymes to carry out their function. Okay, that's the end of part one of this three-part series on microbial growth. Stay tuned for part two, and thanks for watching.